I'm going to tell you about some of the work that we've been doing on trying to understand how bacterial codon usage evolves. I apologize to those of you who are from NCBS. You've probably heard me talk about this a million times. You can go get some coffee. So I think Nick has already introduced the idea of codon bias and why it's a bit of a puzzle and has been a puzzle for a long time. And I'll tell you of some experiments that we've done to try and address this problem and try and understand what are the kind of selective forces that could be causing evolution of codon bias. So just a quick recap, what do I mean when I say codon bias? So here's an example of leucine codons in different species of Vibrio, and you just see pie charts. Each of the colors corresponds to a different codon, all of which encode leucine. Right? And you can quickly see that there's a bunch of species of Vibrio cholerae, a bunch of strains, all of which use CTG as their primary codon to encode leucine in their genomes. Whereas various strains of Fisheri seem to use the CTA codon much more frequently. And these kinds of patterns have been observed for decades now, actually since the 1980s when people first had a little bit of genetic data on different genes from different species. And they quickly realized that there's a lot of bias, and the nature of the bias and the degree of bias varies across bacterial species. This is also true for eukaryotes, but the variation in bias is much less for eukaryotes. So for bacteria, it ranges from something like 15% or actually even 13%. Sorry, what am I talking about? The range in codon bias is much lower for eukaryotes typically than what we see for bacteria. So for bacteria, also because we could do experiments much more easily, it's been much more attractive for me to understand. So how then does it evolve? And there has been a lot of theoretical as well as experimental work to try and understand this. Broadly, you can sort of categorize the ideas into two basic sets. You either have neutralist kinds of arguments where people say that codon bias is an emergent property, that there are some selective or neutral processes that actually act on other traits, and codon use itself sort of just drifts along. There are also selectionist sort of arguments which people have proposed, which focus on the selective benefit or deleterious nature of specific codons in specific contexts in the genome. And of course, none of them is probably sort of the full truth, and there's a mixture of these. And we need to really understand the relative importance of them and the factors that determine the relative importance of these different features. So here's a list of what I think are important things or important factors to understand codon bias evolution. The first one is genome GC content, which sort of one can bin into the neutralist camp initially. People propose that if there is any selection at all on genome GC content, and this selection is driven by mutational biases and differences in mutation repair enzymes, which cause mutational biases across genomes, you might have GC content changing. And people have, of course, observed that there's a very nice correlation between the average GC content of a bacterium and the codons which it uses. So typically, GC-rich bacteria will tend to use GC-rich codons. That seems to be straightforward. And so the idea is selection on genome GC content strong enough to give rise to the kinds of biases we see in codon use? tRNA genes are thought to co-evolve with codon bias. So one of the oldest hypotheses, which was selective in nature, to explain codon bias, proposed that as the ribosome moves along and translates mRNA to make protein, it has to pause at rare codons. And I think this is, again, something that Nick brought up earlier. And this pause is perhaps deleterious because then you're reducing the amount of protein per unit time that's being produced. And so if a tRNA is abundant in the genome, in the cell, it's likely that its cognate codon is going to be used much more frequently. So again, you do observe a nice correlation between tRNA gene copy number or tRNA levels in the cell when people have measured these and the codon use, but it's not perfect, and again, there's a lot of variation there. So we still have stuff to explain. These are sort of more features that have come up in their importance much more recently. So mRNA structure is also thought to be an important feature which can regulate codon bias or codon use. And this is because if your mRNA as a function of the codons that are being used sort of folds really tightly or is very loosely folded or has some structure or does not have structure, That itself could lead to stability of the mRNA being under selection, and then that can feed down into codon bias. Finally, you can have various regulatory motifs which could be affected if you use different codons, right? And so I think it's pretty obvious to all of you that's a fairly complicated picture which we don't fully understand at all. Here's an attempt to sort of tell you what is being done and what we are trying to do in our lab to address some of these questions. So one of the first things we started off doing is trying to understand and quantify how much variation in GC content do we see in the bacterial phylogeny and how is it organized. Can we look at some rates of evolution? So here's just a phylogeny of all sequenced bacterial genomes, and we've colored them based on a simple reconstruction based on Brownian motion of what is the genomic GC content likely to have been going farther backwards in time. Right? So here are all our, at the tips are all our present species which we have genome sequences for. The yellow and green indicate lower genome GC, and the blue indicates higher genome GC. What you see is there are some clades such as actinobacteria which seem to have retained higher GC pretty far back in time, millions of years. And there are some clades which similarly have had a very early shift. This is just hypothesized based on Brownian motion models. A very early shift in low GC content, and that seems to have been retained. And then there are these weird groups of bacteria, most of which are either pathogens, obligate pathogens, or obligate symbionts of various animals, which seem to have extremely low GC content. These are these bizarre examples of sort of 13% GC, right, in their genome. That's absurd. I mean, 1/4 of your genome has GC, that's it. The rest of it is all AT. So, so one question is, well, what leads to this, these kinds of rate shifts, and what are the rates at which GC content seems to evolve? And then eventually we want to answer the question of, can these sorts of rate changes help explain the shifts in codon use that we observe in the bacterial phylogeny? So here's a closer look at a smaller clade within this phylogeny. So here's the chlorobium clade, and a few species here at the tips, again colored according to the GC content. And on the right side is the relative codon use of the CTG codon for a specific amino acid. What you see is that as the codon, this particular codon, gets used increasingly, and it's a G-rich codon, relatively speaking. The alternate codon is CTA, which is relatively A-rich. You see that these match up fairly well, but not always. Right? So here's increased GC content on this branch, and you have an relative increase in the use of the CTG, which is a G-rich codon. As you have a decrease in the GC content in the genome, you have less CTG being used, and the alternative A-rich codon is being used more. So we want to sort of go and quantify each of these instances where there are matches and where there aren't matches, and ask, is there a nice and tight correlation at this level? The other thing we're starting to do is to quantify the rate of GC change across taxa. So here is a subgroup of actinobacteria with sort of marked these different clades, and we said, let's try and quantify all the pairwise differences in GC content that we see in this clade, and plot that as a function of the phylogenetic distance. This distance is based on published phylogeny, which use a large number of, a handful of highly conserved genes and looked at their sequences to identify phylogenetic distance. What you see is that the gray points show you all possible pairwise comparisons across the entire group, and the colors highlight the specific pairwise comparisons within these colored clades. And what you can see is that some of them have much higher rates of GC change for a given phylogenetic distance. And also, the slopes of these are quite different even when compared to the null distribution, which we can generate by scrambling up the tips and the GC contents in that group. And when we can sort of generate these null slopes based on the null distributions we expect if we don't have information on these GC contents. And the actual slope we see is higher than all of the nulls, which are lines here that maybe some of you can't see. I apologize for that. So the point here is that the slopes are different in different subclades, and we don't really understand why. So we're trying to figure this out as well. Yeah. Yeah. So that distance is actually based on the amino acid sequence, not on the, yeah. But that would make it very circular, yes. Right, so I want to move on to tRNA gene copy number. And the point of showing you the previous stuff was just to say that these are questions that we really need to answer and we're trying to answer. And hopefully they'll give us a better picture. Okay, so what about tRNA gene copy number? As I mentioned earlier, people have noticed these nice correlations where here's the frequency of codon use, and here's the concentration of the tRNA which recognizes each codon. And you can see there's a nice correlation. But this correlation does change at different growth rates, right? So correlation is much tighter during exponential growth and it's much weaker during other phases of growth. This is for bacteria. And we know that from in vitro as well as some in vivo experiments that rare codons tend to be translated more slowly. So there seems to be good evidence to suggest that selection on translation rate, which is the rate of protein production, could potentially cause changes in codon bias. This is just a cartoon showing the ribosome, which has a tRNA and is translating a piece of mRNA. And the idea here is that the pool of tRNAs available to the ribosome is really what's rate determining in this process. So to really understand, is codon use even visible to selection at all? Because for a long time, people hypothesized that selection on codons is very, very weak or moderate at best. We wanted to ask, well, how much can we quantify what is this trend of selection acting on specific codon changes? So we did some experiments. We took Methylobacterium extorquens. It's a nice pink bacterium, happens to be everywhere, doesn't do much. But it has this beautiful property of being a methylotroph. What that means is there's very few of these species that can use single carbon substrates as single carbon compounds as a carbon source, such as methanol and methylamine. And so we have a really nice fitness assay for it. And we're focusing on this enzyme called formaldehyde activating enzyme, which is an enzyme that is necessary to make use of methanol and break it down, right? So without FAE, these bugs cannot grow. And if we mess with FAE and we change the codons in FAE, we can then ask questions about what is the fitness impact of codon use. So I'm going to cut a long story short, but here's the main result, which is that the growth rate of strains which carry different synonymous mutations in the entire gene, FAE gene, is much lower on methanol compared to the wild type. But on succinate, which is a multi-carbon substrate, where FAE is not required at all, there's no impact on growth, right? And so what we see is that, yes, there is a huge fitness impact of changing synonymous codons, of making synonymous mutations in this single gene, which is important for fitness. And so, sorry? Yeah, so there's a lot of mutations in most of them. This is a version which has only frequently used codons, and so that has over 100 point mutations in 500 odd base pair sequence. There are some, I think this one here has only about 40 mutations, and the others are all in between. Yeah. Okay, and the reason that we see this growth rate defect is because they're all producing less protein compared to the wild type, right? And so the problem arises because there's less protein being made, but we're not exactly sure why there's less protein being made. It could be translation, it could be mRNA, it could be a bunch of regulatory motifs. So we wanted to figure out what is going on. So we did experimental evolution in the lab where we start with single colonies of each of our mutant strains. We evolve them in batch culture, and at a point where we start seeing that the growth rate of the populations have increased and has reached some sort of plateau, we can say, okay, they've adapted fairly well, starting from a really low fitness. What is the genetic basis of this adaptation? So this is just to show you that if I take different populations, replicate populations, they all increase from their initial growth rate, which is quite bad in some cases, to a final growth rate, which is not too far from the wild type, which is shown in gray here, right? Yeah, don't worry about these right now. So we sequenced the FAE gene for all these populations because we expected that selection would have changed something in the FAE gene, considering that's what we messed with. So here's a cartoon of the first 14 residues of the gene, which is where we found all the concentrated mutations, right? Each dot showed you where a mutation occurred. All of these are synonymous mutations that were found in a given population. Sometimes some of them were repeated across multiple populations. Here are non-synonymous mutations. Here are some non-coding mutations which occur just upstream of the sequence, right? Which could potentially be regulatory mutations, perhaps in the promoter region. These guys, these are too far away to be promoter mutations, but maybe they're doing something. We don't know. Doesn't seem to be because the wild type overproduces it by a lot. Yeah. It is rather poorly regulated. And potentially one reason is that it's hypothesized that this is a horizontally transferred enzyme that has been shared across multiple bacteria in this group. So it's possible that it's just relatively recently transferred, hasn't sort of figured things out with this regulation yet. Any other questions here so far? So the thing that's important to remember also is that a given isolate that I picked up from the evolved population only had a single mutation, right? So I'm showing you all of these mutations here, but no mutant had more than a single point mutation. So, and here's the protein that they're producing. And the triangles all show you one of these mutations, each of them plunked back into the ancestor to say what is the impact on fitness. And you can see that all of them are beneficial, right? Extremely so. So what we have is some of these synonymous mutations are super beneficial. It's just one codon which is moved, which is changed, and that has such a big impact on fitness. So this is, I think, very interesting and surprising. And increasingly, I mean, I think I see more and more cases where people are finding synonymous mutations that they do find in external evolution. Previously, they would ignore them, but hopefully people don't ignore them all so much. Isabel has found some synonymous mutations in viruses earlier. And I don't think you looked at the fitness impacts of those point mutations, but they were likely beneficial because they occurred so many times in repeated instances. Mm-hmm. Yeah. No, so this is one of these mutations which is a non-coding mutation. So it's not one of the coding mutations, right? So we don't know why. I mean, and there's also some error associated with it here. So I'm not very sure that this is actually a very beneficial mutation at all. No, no. These ones are too close to the start, and these guys are too far out. Yeah. Yeah. Any other questions so far? Yeah. So unlikely because we moved all of these mutations back in the ancestral background. So before we started off the evolution lines, we had frozen down everything in the -80, and so we pulled those out and put them in. So what I would need to really answer your question is to sequence those guys. That's kind of too expensive to do. Yeah. Mm-hmm. So the coding mutations were all GC to AT mutations. Some of the non-codings were the reverse, but largely most of the mutations we see GC to AT, yeah. Some were, yeah, well, actually some of the regulatory ones are also deletions, point mutations, which cause deletions. But I think one or two of them might be AT to GC, but they're largely GC to AT mutations. Okay, so what is selection acting on then? It's clear that selection, these are adaptive beneficial mutations that have increased in frequency and have very, very, very parallelly increased. So the question is what is going on there? So here's just a summary from a nice review paper summarizing what are the potential mechanisms at the molecular level for what is selection acting on. It could be that the stability of mRNA is affected by single mutations. This is relatively unlikely in our case because, as I said, a single point mutation does not change the mRNA stability too much, but we can test this. You could have local stability near the start codon, which is particularly important. So one thing that I forgot to mention, kind of obvious, but is that all of these mutations are in the 5' end of the gene, right? So beginning of the gene, which is kind of, not entirely expected necessarily. So why would all of them cluster in this area? Presumably because we had mutations initially made in the rest of the gene as well, right? But none of them were affected. Sorry?
control on the ribosome, you have secondary structure form, you get regulation that way. And you often see that in things regulation.
We don't know anything about FAE really. So, but when we do measure protein levels, they don't increase as much as you would expect, given the amount of fitness increase. So I don't think it is just protein translation levels which have changed and which are responsible for the benefit, at least not in all cases. Some cases potentially, yeah, yeah. Yeah, so there's another hypothesis that says that, you know, if you have rare codons early on, then you have this sort of ramp effect where you prevent traffic jams by having everyone enter your highway a little bit slowly. And if you enter the highway really fast, then everyone's going to bash up against each other. That's potentially what's going on because we see mutations in this region. And another possibility for bacteria especially is that because bacteria translate at the same time as they're transcribing, maybe it has to do with folding of the protein. If it's not correct, it may not end up in the right part of the bacterium, or it may not end up folding correctly. Right? So these are all possibilities. Yeah. So why would AT be easier for polymerases to bind? Yeah, sorry, she's saying whether the GC280 mutations made it easier for polymerases to bind. Yeah, but promoters will be for transcription, not for translation, right? Might. So we do see mRNA increasing to some extent in some of them, but I didn't show you the data here, but not all of them so much. And the correlation between mRNA and protein increase is also not super fantastic. Someone else had a question, yeah. Some have much bigger changes than others, but for the large part, you don't see a nice sort of linear correlation between the amount of increase in fitness and the amount of increase in protein. Not that one necessarily expects that, but you see much bigger gains in fitness relative to gains in protein production. In most cases, but not in all cases. Yeah. Other questions? Yeah, let me try. Not that I have an answer, but and then there was this more recent hypothesis that proposed that one of these regulatory motifs, not quite regulatory, but motifs that might be important, is a Shine-Dalgarno-like sequence in the middle of the transcript. The Shine-Dalgarno sequence is this sort of 6 to 8 bases, which is typically upstream of a gene, which is on the mRNA. And that is what the ribosome recognizes and says, okay, I need to start translation a little bit downstream of this. Right? And so the idea is if you have something that looks like this motif in the middle of your mRNA, it's going to try and recruit ribosomes there to start initiation at an odd place, or it's going to cause ribosomes to pause so that there's again a traffic jam, or you don't make enough protein per unit time, right, or per unit mRNA. And so the idea was that if you have this sort of pausing or internal initiation, either way, you get blockages, and that might reduce the protein production rate. And so maybe that is what selection is acting on, and maybe some of these point mutations we have changed some of these SD-like sites in the middle of the gene. So we tested that. So we calculated or estimated what is the binding affinity of this stretch of DNA that we have, which has had mutations, to the anti-SD sequence, to the SD sequence of this species. And we made a bunch of these point mutations. So we said, okay, let's take all of the first 50 base pairs and make all point mutations in silico and predict what is the impact of these mutations on affinity to SD and for the mean folding, minimum folding energy, which is a measure of mRNA structure. So if MFE is very high, it means the structure is very, very strong, right? And so these are all sort of computational predictions based on models, folding models for RNA that people have, someone out there has developed and thankfully made freely available to the rest of us. And so we can use them to say, well, here is the prediction of, here's the wild type, right? So if the wild type is optimal and it seems to be relatively optimal for us because it's got the highest fitness. What we'd expect is that if we made some of these mutations that are predicted to increase anti-SD affinity, sorry, decrease anti-SD affinity, and get more like the wild type, we should find that fitness should increase, which it clearly doesn't. And similarly, if we're getting more and more like the MFP for the wild type, the RNA structure for the wild type, we should be getting higher in fitness, which also doesn't work really well either, right? So here's a whole bunch of point mutations that we made on plasmids, and we put them in a knockout for FAE, and we tested growth rate, and there's no correlation, which is really irritating and annoying. But all it's sort of saying is that there's no single mechanism that can explain the fitness benefit of all of these different mutations, right? And it's sort of, one doesn't expect it necessarily. Why would you, right? So it's only when you look at it from the lens of codon bias and how codon use has evolved that one expects perhaps one mechanism. But no one in their right minds would show you a bunch of non-synonymous mutations and claim to have found one mechanism that is responsible for all of that. So in some sense, it's not surprising, but it is sort of annoying that there's no answer and there's no mechanism that I can tell you. Yeah. Yes. Hmm. Hmm. So what we're measuring is the rate at which that increases. So that is the growth rate. So the absolute turbidity is not being looked at here at all. Right? So we're looking at the slope of the change in turbidity per unit time. And that is what we're measuring as the growth rate, right? So even if the actual turbidity is changing, it won't make a difference to what we're measuring. Does that make sense? Again, it's the rate of change of that turbidity which we're measuring, right? Maybe I can describe or explain to you later. Okay, and so the other interesting thing was that we see this massive parallelism where in this strain, which has only frequent codons to start with, we see one single synonymous mutation that shows up in 9 replicate populations, right? So it's super parallel at the nucleotide level, and that's very, very rare to see in experimental evolution, as far as I know. And we wanted to see, well, what is with this huge parallelism, right? How come we don't see any other mutations show up? Because clearly those mutations were possible in this strain, but they only occurred in a different strain. And so we made some of these cross mutations. That's what I'm showing you here. So the triangle is what we actually saw evolve in this strain. And the black circles are evolved mutations from the other alleles, which could have occurred here but did not occur, right? And when we add them in, we see that only in very rare cases do we find that these filled circles, which are other mutations that could have occurred, most cases they're much lower, they impart a much lower fitness than what actually evolved, which is the open triangles, right? So potentially, what did evolve in each of these lines is one of the most fit mutations that was accessible to this strain, right? Given the specific sequence that that allele had. Yeah. Yeah, so he's asking, what if you don't, you grow these things in a media where you don't need this gene? So we haven't done it for these particular strains, but well, actually we sort of have. We haven't measured it, but before we do these measurements in the presence of methanol or methylamine, we normally grow them up in succinate, which is permissive for everybody. And I don't think we've noticed differences there in that initial growth phase. But what we did do is quantify the growth rate in succinate, which is permissive, for all of the evolved strains. And there everyone was equal on succinate. And so also what has not happened is improved ability to grow on methanol, given this problem of FAE, has not resulted in a decrease in the ability to grow on succinate. Yeah. So we don't see any real evidence for that. Okay, so just to sort of summarize, what are the questions we're asking now is How often does genome GC undergo major shifts? Are they associated with ecological shifts? So you sort of stare at these phylogenies and stare at some of the ecological data that people have sort of again put up for a lot of these bacteria. It seems that sometimes major sort of really fast shifts in GC content might be associated with pathogens or symbionts evolving in a short time period. So we want to quantify that and see whether that's very common. The other question we're asking is, are these sorts of shifts associated with major gains or losses in DNA repair enzymes? Because these may be causing major shifts in the mutation spectrum. Right? So we know that, people who've worked with bacteria for decades have shown that if you knock out some of these basic repair enzymes, you can change the spectrum to be from GC to AT biased, flip, or cause a bias where none existed, and so on. So we're asking, if we make some of these changes in the lab in experiments, do we see changes in GC shifts? On the flip side, on the phylogeny, can we map the presence and absence of these repair enzymes and ask if that's correlated with major shifts in GC content? So we're doing that. So the other question, like I said, we're asking whether DNA repair mutants can drive major shifts in GC content. And the very first question to actually ask here is, can these mutants even establish, right? So if I make a repair knockout, and we know these natural lineages exist, so all repair enzymes that we know of are not present in all bacteria, right? So clearly they can be gained and lost. Their gain and loss does happen. We don't know what are the impacts on fitness. So Mridula, who's here in this course, is doing these experiments where she has mutants that have been knocked out for different repair enzymes, and they are predicted to give shifts in the mutational spectrum. And so now she's evolving them under drift or under selection and asking, can these mutants establish, given a wild type situation with which it's competing, can these mutants actually increase their fitness? And does the fact of having a biased mutation spectrum actually give them any fitness advantage at all? And then what are the conditions in which these could be true? And finally, we're trying to map tRNA gene copy numbers and codon bias and GC content on this big phylogeny and say, are these changes correlated? And eventually, can we start going from correlations to being able to infer causality on the phylogeny? So that's where we're going. These are all things that I think are very, very tightly correlated, and we don't quite know how and why. But we need to understand these on a phylogenetic framework. And these are the people that I'd like to thank for all the work. So Kritika, Mridula, and Gaurav are all PhD students in the lab who've been working on this codon bias project, which I brought here from my postdoc, but they did a lot of work to finish it. Chris is my postdoc advisor, but was, with whom I started this work initially. Saurabh is also a PhD student who was working on the phylogeny of and mapping GC content. Nachi was a JRF who did a bit of the work for the phylogenies earlier, and Alefia was also a JRF who helped with the Quran project. Thanks to all these institutes for funding. And any more questions? All right, coffee. Yes. Oh, sorry. A couple quick things. So there's coffee outside now, but we can start posters right away. The poster session technically ends at 6:30, but go on as long as you want. And dinner is going to be right there outside where the posters are today. It's like a special dinner of some sort. So stick around and enjoy yourself. And the shuttle.